Okay, I'm grading your exams, so um, depends on who you are. I'm trying to be fairly generous. First exam. So, what was your? You can be very frank and honest with me. What was your sense of the exam? What was it? Short. Okay. You wish there were more questions? Okay. Does everybody agree with that? Okay. More multiple choice? So here, and this is, I know that there's always it depends on the student, but a lot of times my exams kind of tend to be a little shocking to some people, okay? So a lot of times I'm just not asking you to like memorize stuff, and so I'm asking you to kind of think about things or put things together, and I think, so is that? Okay. Okay. Okay, so I'll make it harder. No, so here's what I'm going to do. I'm going to You'll get your exams back, and you should look at them, and I have all of your attendance grades, I have your homework, I have your lab grades, and your first exam. I'm going to kind of give you a general idea of how you're doing so far, and if I think you're struggling a little bit, I'm going to reach out to you, okay? And we're going to try to provide some help for you if you need it, okay? So I know that sometimes, so we have a peer instructor that might be able to help you, or you can come in and see me. So so again, this is a course where you can drop one of your lowest exams and you can take the final to substitute it. So yeah, it's not totally cumulative. It's about half of the material will be on the final. So if you did not so well and you're a little worried, that's always an option to kind of recover. Okay? The other thing is inquisitive. Do you want me to extend that deadline? It's a lot. Just get to 75% and stop, right? Easier said than done. Yeah, it's hard. So I'm going to extend the deadline until tonight at midnight, and I'll send an email out. So if you want to do it or not, it doesn't matter. You can get 2 extra credit points for each module if you score 75% or above. So I'll send, so you can try to do that. And then I'll return the exams. I'll probably post the grades, I don't know, over the weekend maybe if I get to it. So, and then I'll turn them back on Tuesday. And I'll post the key on Moodle. Okay. Yeah. Huh? Yeah, the deadline, I'll extend it till tonight. So, okay, midnight or something, or, Friday morning I'll check it and start. I got to start adding the extra credit points to your exam sometime soon, so. Okay. Any questions about that? Yes. Yeah. Yeah, you get that. So there are also 3 additional extra credit points from the in-class assignments. So I've tallied all those up. I pretty much, if you turned it in, I gave you points. Okay. Yeah. No. So somebody asked me, oh, if we do the reading quizzes, can we get extra credit? And I said, well, sorry, no. Just kind of use that as a way to kind of look at questions. The other thing to remember is inquisitive and the reading quizzes cover a lot more material than I cover in class. So there are going to be topics in there that you're going to say, that doesn't make any sense to me. I've asked the publisher if there's any way I can create reading quizzes and delete questions that have nothing to do with what I lectured about, but they don't give me that option. Okay? So I would just, as I go through those reading quizzes and you see something on coalescent theory and I've never talked about it, I would not try to answer that. So I would just focus on the kind of questions that sound kind of familiar. Is that it? I think it could be helpful. Okay. Okay. So we're going to do a little exercise today midway through the class for a couple extra bonus points. Not for the second exam. So, okay. So what we're going to do today, and I'm not talking a lot in the chapter 9. There's a lot of stuff on what's called linkage disequilibria, all this sort of stuff. I'm not going down that path, okay? So what we're going to talk about today is evolution at multiple genes or multiple loci. And the reason this is important is we're going to be finally moving away from A1 and A2, or big A and small a and big B and small b, and we're going to be moving towards what I think are more interesting things, to look at, which are what we call quantitative traits. And these are traits like height, fruit size, bill length, bill depth, all these things that have what's called continuous variation, and we're going to be looking at how those phenotypes are influenced by both genes and environment, and how they evolve by natural selection. And so, and this is going to move kind of directly into population differentiation, speciation, and all these things. So what we're building on are the concepts in the first part of the course are building into sort of these evolutionary processes and how species formation occurs. So what we're going to look at today is how do multiple genes interact. Often multiple genes interact to determine phenotype, especially in traits or characters that have what we call continuous variation, not discrete variation, which is red or white, but like height, color, all these things often have sort of what we call continuous variation. And we'll be spending a lot of time looking at continuous variation, and we normally characterize those in terms of a distribution. Any of you guys had stats? So you know a little bit about normal distributions, means, variances, standard deviations. We're going to be looking a lot at sort of average values, variances, and standard deviations of these continuous traits. We're then going to look at what are called adaptive landscapes and how they can help us understand the course of evolution, and especially species differentiation. And what we're going to be looking at is how we can look at how traits change in what we call an adaptive landscape, and how that can be influenced primarily by selection, but drift often plays an important role. And then next two things we're going to use what are called quantitative genetic models, and we'll talk about that, to predict changes in traits, even when we don't know the specific genes that are causing the variation in the trait. And when we look at quantitative genetic models, what we're going to say is whatever phenotype we're looking at, if it's plant height or flower size, right? That's usually what we call a quantitative trait. We believe that both genes and environment influences the expression of those traits in individuals, so we'll be doing that. And then the last thing we'll look at is how does selection affect the mean and variance of these quantitative traits. And we'll be looking at 3 different kinds of selection, and that last slide will be kind of an important one. So, okay. So let's look at this, and this is a very common phenomenon when antibiotics are used. And this is a sulfonamide that is used to treat a pathogenic E. coli. And so if you look at this, this is a common pattern that's seen. So here, starting in 1987 up to 1999 to 2005. Along this y-axis is the sulfonamide prescriptions per year that were distributed. I think this was in Britain, where this study was done. And along here is the percentage resistant to sulfonamides. What do you notice here in terms of what happened? So what antibiotic was used to treat this E. coli in Britain starting in 1987? Well, this particular antibiotic. And then what happened to the use of it? It declined, so that by basically 1997 or 1996, doctors were no longer subscribing this antibiotic. And why? Because it's resistant. It developed, as we mentioned, fairly quick antibiotic resistance to this particular kind of antibiotic. So what you'll notice is that the percentage that are resistant to these sulfonamides, we didn't track it from the beginning, but it was probably fairly low. But by 1991, basically 4 years later, what happened to the percentage of these E. coli that were resistant to this antibiotic? It went to 40%. Is something surprising to you beyond that? Yeah, wouldn't you think that this would go down? Okay. That's possible. Huh? Yeah, a little bit. So after they stopped so they continued the antibiotic use, and it went up, it went up here, right? But then it declined a little bit. But it's kind of surprising that it didn't decline a whole lot more when that, So it seems that resistant gene in that E. coli population, even when the antibiotic is not around, right, is still being maintained. So, yeah, well, that's one thing you might want to do is this was, what, 1999? By 6 years later, it dropped a little bit. It might be interesting to know what's happening now. I suspect there's a further decline, but it didn't go to really low values. And this is a common phenomenon that happens when antibiotic resistance occurs in bacterial populations is even after the antibiotic is no longer used, they still maintain resistance to it. Okay? So, and this is one of the big problems with, and I'm not saying we shouldn't use, don't get me wrong, I'm not saying we shouldn't use antibiotics and we shouldn't develop new ones, but we might want to think about how we use them and what kind of combinations we use them in. Okay, so what do you think? What could explain, well, the thing's not around anymore favoring the resistance gene, why isn't it lost? Can you think of any reason? And again, now you know these are the sorts of questions I ask on exams, right? Can you think of anything? That's a possibility, okay? So maybe there's another mutation, or maybe, and as you mentioned, maybe that resistance to that particular antibiotic is also resistant to the other antibiotics. So maybe there's a widespread, maybe that gene confers more resistance to more than just that antibiotic. So that's a good possibility. Right. Yeah, they treat, obviously when this went down, do you think the doctors in Britain quit treating diseases? No. So they do, obviously another antibiotic was used, and so maybe it had, that's another possibility. Yeah. So what's another possibility, though? Well, maybe having that resistance gene doesn't have much what we call a cost. Okay, so let me just, here's the answer. Here's what happens. Okay, so again, let's look at, this is kind of what you start out with. So this could be a bacterial genome, bacterial genome, E. coli, for example. This is probably the ancestral condition in which you, basically now we're not just looking at one gene, we're looking at two genes. One is involved in resistance and one has to do with what we call compensation, and I'll explain what that is. So what we have, at least initially, is this is the genotype at two loci, small r, small c, and it's shown down here, okay? In the absence of an antibiotic, right, what's its relative fitness? It's 1. It's set at, that's the highest, okay? What's its relative fitness when there's an antibiotic being prescribed against it? It's really low. It's not resistant to it, okay? So that antibiotic early on kills that bacteria. Then what's the next step in the process? Kind of an equilibrium, but as you go to here, what evolved? Yeah, big R is what we call the resistance gene to that particular antibiotic, whatever it is, okay? This hasn't changed yet, and we're going to eventually get to that, okay? So we imagine that a mutation, is this a beneficial mutation? Okay, so this was a mutation, arose at random, but it's beneficial, it confers resistance. There are lots of ways bacteria get this, often from transfers from other organisms. There are lots of different ways that these resistance genes pass between things that are quite unusual and may not involve mutations, but just imagine a resistance gene arose, okay? Now let's look at the fitness of that genotype. In the presence of an antibiotic, what happened to it? Its fitness went up, okay? Quite a bit, okay? And here's the relative fitness of this thing when there's no antibiotic. What happened, though? When there's no antibiotic, having that resistance gene, what did it do? It actually declined. Overall, it's in the absence of the antibiotic, the fitness of this resistant bacteria actually declined, okay? So it went down. See here? Down, up. So the resistance gene had some effect, right, in terms of it conferred an advantage, but in the absence of the antibiotic, it actually reduced the fitness. That has some consequence. It might have slowed its replication rate, division rate. the presence of that gene had some other effect besides just antibiotic resistance. And usually it's on growth rate. It slowed it down. It slows it down. Okay? Now what? What happens next? What other gene changed? Yeah, the C, this is what's called and you don't have to worry about it. This is just called a compensation gene. And basically what happens is it mutated from the small c to the big C. So now the final genotype is big R, big C. It's resistant and it's compensated. What's it doing? What's that big C? What happened? In some sense, yeah, it's kind of making it, yeah, more resistant, but it's kind of making it just improving its fitness, right? And so the idea here is that, especially when you look at no by antibiotic, it happened in both, but let's look with no antibiotic. What happened is it compensated for the presence of that other gene that kind of had an effect on it, so it was able to increase maybe its growth rate, its replication rate, how quickly the cells divide, you know, and those sorts of things. So the idea here is that, and what I'm trying to introduce is that two genes can influence the fitness under antibiotic resistance. And this is extremely common, that even when you have that resistance gene, right, and it kind of has initially an effect on fitness, it's compensated for. And actually it turns out that it gets pretty close to 1, doesn't it? So, yeah. Right here? Yeah. So why did this go down? Anybody want to tell me? Yep. The big R gene, right, actually caused a reduction in that growth rate, independent of whether it's in the presence of an antibiotic or not. So that's actually the effect of that gene. Or, you know, the resistance has an effect on its growth rate. It had a negative effect. The evolution of resistance has a consequence on the growth rate of the bacteria. Does that make sense? Okay. So this is, you know, so again, so if I were to ask you, does this explain this? Does this explain why this resistant bacteria doesn't decline too much? It has a pretty high fitness, right? It's at that compensatory mutation. Even in the absence of that antibiotic, right, it still has a pretty high fitness relative to the ancestral one. So it's kind of really hard to get rid of that thing. And this is but it also could be the other thing. It could be resistant to other antibiotics. But the idea here is this doesn't decline too much because at the end of this evolutionary sequence, going from here all the way to here, this compensation allows these things to have, you know, it's not exactly 1, but it's pretty close. So it does pretty well. Does that make? Okay. You look perplexed. Okay. It's a little bit confusing. Okay. So I'll give you a little work problem. We're actually going to be doing this in a bunch of other cases, so I think I'm going to force feed you this. Okay. So just think about that. We're going to come back to the little work problem, the little group problem you're going to work on is very similar to this. I'm going to give you 4 genotypes at 2 different loci, and I'm going to ask you what should happen. So, okay. So what we're going to be looking at today are what we call quantitative traits. Instead of just red, pink, and white, or, you know, tall or small, tall or short, we're going to be looking at what we call quantitative traits. And let's just start up here. Imagine the parental generation. Now we're not just looking at 2 loci, right? How many do we have here? How many genes do we have here influencing this trait? We have 3. And in fact, most quantitative traits that we look at, whether it's plant height, all sorts of continuous traits, anywhere from 6 to 20 genes can be involved in influencing that particular trait. Okay. So here what we're doing is we're crossing, this is a triple homozygous, right, with another triple homozygous individual. And what's in the F1? they're going to be all double heterozygotes at each locus. And then you cross that, and I'm going to make you do a Punnett square with 5 genes on the test. No, I'm not. That's why you never do Punnett squares. Once you go above 3, you do what are called path diagrams. So anyway, you'll do that in genetics. Okay. So here's a Punnett square. I'm not going to ask you to do this. What it illustrates is a point, is if you cross 2 parents that are heterozygous at all of these 3 loci that influence kernel color, okay? Those look like lips to me. Okay. That's what I was going to say, lip color, and then I read the thing. Okay. So these are kernels. These are, yeah. Look like coffee. Okay, they're coffee. So red kernels are, this would be the most extreme phenotype, and this would be the most extreme phenotype at the other end. Red kernel individuals crossed with white kernel light individuals produce F1 offspring with intermediate color. And then when you F1 offspring, all triple heterozygotes then cross to produce a range of color variants in the F2 generation of the offspring. What do you notice here? There's a lot of variability in kernel color, isn't there? Okay? And basically what we're saying, we basically said these are what we call our additive genetic effects. So I'm not going to go through all of this. I'm going to kind of go through this part, this diagonal, and down here. Okay? Here are, when you think about it, here's the most extreme phenotype, isn't it? In terms of dark red coloration. And that individual right there is the darkest red. It's homozygous for the cap letters, right? If you look what happens here, these are all, this offspring is all the same with this one except for what? There's a small c there, right? So that offspring is going to be big A, big B, and big C, small c. There's one allele that's different, right? And is it exactly the same color as that? It's slightly reduced. And this is what we call basically what's called an additive effect. Okay. Let's go down to this extreme. So this is the other extreme of the phenotypes in the population. What do we have over here? This is homozygous for all the small caps, right? And what's its phenotype? It's the lightest, okay? And you can do the same thing here. This is still kind of whitish, but not as white, okay? What's right in here? These things are all the same color, right? I think, should be. They're right on the diagonal. Guess what they are? They're all heterozygotes, right? With various combinations of the alleles. And where do they fall in the distribution of color? Kind of right in the middle, okay? So what we're saying here is kind of simple, is the individual phenotype, and the phenotype we mean is the color. Here's extremely red, extremely white, and this is kind of gradations, you know, slightly less, right? The individual phenotype is simply the sum of the effects of each allele that it carries, okay? And this is what we call additive effects, and we'll be coming back to that. So, for example, more alleles for dark red color that an individual carries, the darker the kernel's going to be, okay? With the most extreme being here. These are pretty dark too, and those sorts of things, okay? So it's basically the number of sort of homozygous dominant copies you have, the darker coloration you're going to be. Here you don't have any, and you're extremely light. Okay. We're going to come back to this. This is kind of an important concept when we think about quantitative traits. Okay. Let's think about what happens when you have these kinds of traits. So that kind of did the cross. Now we're going to, again, expand it to not just the individual, you know, a cross between 2 parents, but how does this then extend to the population level? Okay. So I'm not going to spend a whole lot of time on what caused this, but let's look at this. What's this thing look like? Is that, what kind of distribution, what would you call that? That would be a normal distribution, okay? And what we're looking at is you have a frequency of individuals, right? This is 0, and this would be, whatever, 40%, whatever it is, doesn't matter. Along here is the quantitative trait. In this particular case, we're looking at height. So again, it's not just small individuals and tall individuals. It's smaller, intermediates, kind of small, kind of tall, extremely tall. So you have this sort of continuous distribution. What are the characteristics of a normal distribution, how we characterize it? And you'll be doing this in lab. But you probably had statistics. You did this, right? Yeah, this is a bell curve. What are some things that we can use to characterize it?
So the middle is...
Yeah, what's this thing right here under a normal distribution? What would we call that? That's going to be the mean, right? The mean height in that population, okay? Or the average height. And you can determine that. And then you have some measure of the variability or variance around that, don't you? Okay. So you can characterize either the standard deviation or the variance, which is a measure of how variable the height or these height phenotypes are in a population. What happened here? So what we did is on this initial population, we said we're exposing it to selection. Here is many generations later. What are we selected for? Or what is natural selection? Whether it's artificial selection or it's natural selection, what individual phenotypes were selected for in this case? Yeah. So obviously what this would mean is these individuals out in here in this part of the distribution have a higher fitness relative, say, to these over here. Okay, so selection favored what kind of phenotypes? Larger or taller individuals. What's changed here then? How is this distribution different from this distribution in terms of those two measures? It's narrower, right? Has anything else changed? Yep. So here, obviously, the mean has shifted from here to over here. So the average value of the trait has increased due to selection. What's happened to the variability of the trait? It's reduced, okay? You can see this has a much broader distribution. This has a much narrower distribution, so the variability of the trait has been reduced. And we'll be coming back to this. Your book talks about this, and you'll be perplexed about how they came to that conclusion, so we'll just leave it at that. Okay? It turns out a lot of times you can get what are kind of unusual characteristics, but they can lead to, that looks kind of odd, doesn't it? How you can go from a population that's like this to variation that's even outside of the original range, but we're not going to worry about that. Okay. So we're going to come back to that. We're actually going to talk about how we measure what's called the heritability of those traits. Another thing we want to talk that's really important, we got to come back to the mice, right? This is what's called epistasis. And so this is, don't you get like keywords you can do in flashcards? Yeah. So type in epistasis, see what the definition is. Okay. Epistasis usually involves that one gene influences another gene and often can override the effects of another gene. So in this agouti example, I'm going to just show you just what this is. So there are two genes that influence coat coloration in these mice in the southeastern United States. One is the agouti gene, and the other one is the MC1R gene. And we're just saying these are each of the genotypes at each of these two loci and two genes. Okay. So in the mice that are DD at the agouti locus, right, what do you notice? The different MC1 variants all produce what? Dark coloration, the exact same phenotype. So what this is saying is the mice here, if you have this big D mutation or genotype at the agouti gene, what does it do to the expression at the other gene, which influences the coloration? It overrides it. So it controls or masks the effect of the variation at this other gene. This is what we call epistasis. Okay? now let's look at I'm not going to go through all of this. Let's look at this example where the agouti gene has the LL combination. Is there much of an effect there? No. Well, I mean, not as much. What you can see is in the mice that are DL or LL at the agouti locus, the alleles at the MC1R locus influence the phenotype. This genotype doesn't override those other ones. So if you have this genotype at agouti and your LL at the other gene, you're a sand mouse, okay? You would be that. That's the genes that were selected in the sand mice. Yeah. No, nope. No. Okay. I should talk about epigenetics, but yeah, it's a little different because epigenetics often involves sort of non-coding or messenger RNA influence. These would be nuclear genes that aren't really what we call epigenetic. Okay. So yeah, those are different. Okay. So here, and just to illustrate the point, one gene can override or mask the effect of another gene if there's what we call epistasis. And this is a pretty common phenomenon in a lot of things. Okay. And here's the take-home message, because the effects of the alleles of the MC1R locus depend on their context, namely which alleles are present at the agouti locus, there's epistasis between these two. Okay? So the expression of this gene that influences coloration is strongly dependent on this other gene. Okay. So now we're going to shift a little bit, and we're going to move to what we call adaptive landscapes. And I'm completely glossing over linkage disequilibria in the textbook and just ignore all that. Okay. And what we do in adaptive landscapes is we look at combinations of phenotypes, right? And we're eventually going to get to the point where why do certain combinations of traits evolve in species and how that eventually leads to speciation. And we'll be going through a couple examples of this. Okay. So here are the two traits we're looking at. One is what's called the inflorescence plus peduncle length. So this is a flower, right? Each one of these dots represents the average value of a maple tree, okay? So there are lots of maple trees in the south. There are a lot up north. There are about many maples, okay? And each one of these dots represents the average value of basically the inflorescence or flower, which is from here to here, plus this little thing that's called the peduncle. It's basically kind of the average flower and peduncle length. Along here is sort of the leaf plus petiole length, okay? So the two traits we're looking at are sort of flower and leaves. And what you'll see here, what does it look like? So each one of these, I think this is just North American, each one of these dots represents the average leaf length and the average flower length for maple species in North America. Is there a relationship here? Okay. So this species down here has what? Very small leaves and very small flowers. Okay? At the other extreme, what do you have? Extremely large, long leaves and extremely long or big flowers. Okay? But there tends to be there's kind of this positive relationship between the two, right? So that as leaf length goes up, what happens to inflorescence length? It goes up. Okay, so that's called a positive correlation. So they might be related to one another. Okay? What do you think influences that? Yeah. So a lot of things we might want to think about is probably a lot of it has to do particularly where these species live, right? Okay? What kind of, where they live in the United States, what kind of environments they live in, okay? The growing season, how much water availability there is, nutrient availability, all these sorts of things, and there might be some, things that live in the southern United States might be very different from the things that live up in southern or mid-Canada, right? Or a maple that lives out west might be very different from experiencing very different kinds of environmental conditions, and selection might favor different kinds of combinations of these traits. So that's kind of, these are what we call combinations of traits lead to into the idea of what are called adaptive landscapes. And Sewell Wright, a famous evolutionary biologist, is the first person who ever developed this. So these are just kind of diagrams. All of these are from your book. And again, what we're looking at here is we're always looking at two axes along here. Now it's not just two-dimensional. Is this a three-dimensional graph? Okay. So here we have leaf length and inflorescence length, same as before. Okay. What's the other axis? Fitness. So, and what's down here? If you're at this part of this curve, what's your relative fitness? Pretty low, right? Where's the maximum? Right at the peak. So these are what are called adaptive landscapes. They're always three-dimensional traits. Two of the axes involve the phenotypes or the traits. The other axis is fitness. Okay? And what we're getting at is, so a fitness maximum is reached at this, what we call, this is the optimal phenotype. That's the combination of leaf length and inflorescence length that leads to highest fitness. And where does that, it's kind of hard to see. It'd be nice to, I should have a rotating graph so you can view this in rotation so you could really fully see it. And this is one of the problems with a two-dimensional representation in some sense. But where do you think that peak is in terms of where we are on these axes? It's pretty much right at the, kind of right in the middle, right? So basically what this is saying is that those with medium inflorescence length and leaf length, basically kind of intermediate, that is the optimal phenotype, that confers the highest fitness. And that's probably unique to that particular environment or those sorts of things. That's just one population or one species, right? Now we can extend it to potentially some others, okay? And so what do you notice here? What's different about this adaptive landscape that's different from the top one? Now we have multiple populations, possibly, okay? So a population might be, might be the adaptive landscape, and here's actually the cluster of points that's found in that population, okay, or species. We're going to extend it to species eventually, okay? So that's a fitness maximum. Are there other fitness maxima, kinda? Yeah, well, there's one here, but it's not as high as that one, is it? Here's another one in this other space, right, of fairly low, small leaves and small flower sizes. Here, what's this one up here? That's a small leaf with a big flower, okay? So here we can kind of see there are 4 peaks in this adaptive landscape. Are they all at the same height, though? Which one's, that's the highest, right? Which one's the lowest? Right, one right in back, okay? So now keep that in mind. Now we're going to try to figure out where does evolution take us if you put yourself somewhere in this landscape or this, what we call a matrix. Where do you think it's going to, yeah? What do the peaks represent? Yep. These would represent maybe 4 different populations, okay? Or 4 different species. The maxima, that's the highest fitness for that particular population in terms of the combination of those 2 traits, okay? So there are 4 of them, okay? Where do you think this is going to go? Yeah, it depends. So let's imagine, okay. These are what are called, what we showed, and they should have put the same graph in, because they had to want to confuse you, okay? Okay. Again, we have 4 populations, or 4 fitness peaks. let's say you start here. Okay? This is the original population. Where do you think this thing is going to go? So a large population typically myopically moves up to that peak. So it basically, it goes from here all the way to that, what's called a local fitness maximum. What's the best place to be in this entire graph, though? The highest peak. And that's called, that's called the global fitness maximum, because that particular peak is the highest of all four, okay? So a lot of times what happens in large populations and when there's no drift, what happens is just kind of selection overwhelms everything, right? And it just goes to there. And I'm not going to worry. A small population, actually, a lot of times what can happen, though, a lot of them kind of drift. And they can kind of get down in here, and then guess what happens? Where do they go? Yeah. They can kind of drift across these fitness valleys, and they kind of get close to that, and then they kind of just boop, 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 they go all the way up to the top. Okay. I'm not too worried about you understanding all of those concepts, but the idea that usually what happens is if you start out a population with certain values of trait 1 and trait 2, it'll basically usually go to the local fitness maximum, especially when it's selections just occurring. When drift can occur in small populations, you can get kind of weird things, and things can kind of move in a lot of different directions. Okay. And I'll actually be talking about a paper just, remember those cichlids in Africa I was telling you about? Remember those? Where there are like 300 species. There's just a paper in Nature that said there are 350 species of cichlids that evolved in the last 40,000 years because it actually 2 species hybridized, and it produced all this genetic variability that then led to this adaptive radiation. So we'll eventually come back to that. And it's kind of related to that. Some weird things can happen that can result in flourishes or moving up to the, away from this peak. Or if you started here, where would you go? You'd go up to there. Or if you started in here, you'd go up to there. But, to get to this global fitness maximum, sometimes some weird things have to happen. Okay. I'm going to talk about one more example that illustrates this, and then I'm going to give you a thought problem to work in groups on. Okay. Does this look weird? What do you notice? Yeah, snakes with a lot of different colors, and also whether they're striped or kind of spotted. These are kind of spotted snakes, and these are more stripy, okay? And so Butch Brody is a guy who's worked on these snakes. He actually collected all these snakes, he marked them, and then he went back and looked at how well they survived. Guess what what are most snakes trying to deal with? Avoid being eaten, right, by a predator. So what he was interested in is how these, not in this particular, it's not necessarily the color variation, but whether they have stripes or they have spots. And he was interested in how they do. And it's kind of weird. The snakes that have stripes have a very different kind of escape behavior. And these are different. I think these are different. I think they're all the same species, different phenotypes within a species, okay? And these that have stripes have a different escape behavior from when they're when they're under predation attack. They have a different kind of escape behavior than the ones with spots do. And he was actually able to determine how these two combinations of traits, one is a behavioral trait, right? How you move away from a predator, and the other is sort of the stripes or spots. So these are the two traits, okay? So let's you really need a rotating graph here to really see this. Okay, so we have 3 axes again, right? Here's whether they're striped or spotted. Here's the most very striped, extremely spotted. And the things that snakes do when some of the snakes do frequent reversals. So when they're on the ground and there's a hawk about ready to, they see it or they sense it or there's some other weasel or something coming after them, right? Some species do frequent reversals. So their escape behavior is not just in a straight line. They're going to go this way, this way. They kind of do these frequent reversals in their escape path, okay? So these are kind of the different traits. So some of them do frequent reversals, some do very few reversals, and basically they're on the forest floor or on the beach and they're just taking off straight, okay? They might occasionally switch direction, but it's pretty much a linear straight movement. They're not doing a lot of these reversals. Okay. So he was actually able to determine the fitnesses of, and there's a lot of variability in these snake populations, and he had the behaviors and he had the phenotypes, and he was able to figure out what their relative fitnesses are. Okay, so what are the fitness peaks? Here's one. Here's one. How's that one? That's terrible. That one's slightly lower. So those are the two fitness peaks. Okay? So let's just look at those. This is, what do striped snakes do? They go straight. So striped snakes have a high fitness when they don't reverse. What about the spotted snakes? That's their fitness peak, right? So here are the spotted snakes down in here, and they have frequent reversals, okay? So high fitness spotted snakes that reverse, okay? So what all this really says is which one does the worst? Yeah, the striped that does frequent reversals, okay? This one's not so bad, but it's lower. These are the spotted snakes that do few reversals, okay? So presumably, what happens to these kinds of phenotypes? They get eaten, right? So they presumably, that strategy, that behavior, given that phenotype, in terms of the spotting or stripes, that escape behavior is not so good. So what would you expect? It's kind of hard to tell, but what it really suggests here, do you kind of maintain both the coloration pattern and the behavior? Yeah, it actually turns out these fitness peaks are actually pretty close to one another. So they actually, you always have spotted snakes that reverse, and then you have striped snakes that don't reverse. And you kind of maintain both of these kinds of phenotypes in this snake. And these are in the Pacific Northwest. Okay. Okay, here, I want you to work in groups. Okay. So here are fitnesses of 4 different wolf phenotypes, right? That have to do with how aggressive you are and the size of their teeth. Okay? And what do you know about wolves? Are they social animals? Okay. They often are in packs. There's often certain kinds of dominance hierarchies, and not only male dominance hierarchies, often female dominance hierarchies, that certain kinds of behaviors might be favored, and so, certain individuals might want to employ certain behaviors, others may not want to. Okay. Here's the first question. I didn't write this out. Where are the two highest fitness peaks and what are the 2 lowest fitness peaks? That should be easy, right? So what are the 2 highest fitness peaks in terms of the 4 genotypes shown there? Okay. And just briefly explain why these 2 that have, well, I've already told you the answer. Okay. Why do these have low fitness? Pick out one of these 2 and tell me why do they have low fitness? Kind of give me an explanation. Okay. Okay. So we'll do that. I'm actually going to ask another thing, but once you're done with that. So work in groups. I'll give you, shouldn't take too long, should it?